Welcome, everybody. Thank you so much to the last T-CAIREM trainee rounds of the year. It's been an amazing year. We've had a lot of excellent speakers, and we have our final speaker today, Sunho Lee, and I will tell you about him in just a moment. Before I do, I just want to remind everybody about the Temerty Center for AI Research and Education in Medicine. We are made possible by a generous donation of the Temerty family, and we're an interdepartmental center that serves as a focal point for healthcare providers, trainees, researchers, computer scientists, engineers, and industry to advance health through AI and technology advancements. I also want to just acknowledge the land at which we are operating. The University of Toronto operates for thousands of years on the traditional land of the Huron-Wendat, the Seneca, and the Mississaugas of the Credit. Today, this is still the meeting place and still home to many Indigenous people from across Turtle Island, and we are truly grateful to have the opportunity to work on this land. So without further ado, I'm going to introduce our speaker. Sunho is a certified gastroenterologist with a special interest in inflammatory bowel disease. He's further trained as an advanced IBD fellow at Mount Sinai Hospital, and during this time, he completed his PhD at the Institute for Medical Science here at the University of Toronto. He has been part of the Crohn's Colitis Canada GEM project research team, and this is a project that aims to understand the preclinical phase of Crohn's disease, and he uses his biostatistics and bioinformatics skills to explore the interactions of genetics, gut microbiome, antimicrobial immune response, and the gut barrier function, and how this relates to the future of IBD. So I think we're going to hear more about that today. And without further ado, I'll pass it on. Thank you very much for being our final presenter this year.
Thank you, Laura, for the kind introduction. Thank you, T-CAIREM, for giving me the opportunity to speak today. Let me share my slide. Okay. Can you see?
Yeah, it looks great.
Okay, let me just. Okay, so thank you for the introduction again, and to the committee as well for giving me this opportunity. Today, I'm gonna show how and why I used machine learning to answer a very interesting research question in the field of inflammatory bowel disease. And the question is, does the microbiome composition predict future development of Crohn's disease? And just to give you a background of what inflammatory bowel disease is, for those who are not familiar with this disease, inflammatory bowel disease is a chronic inflammatory disease that involves the gut. It has a relapsing and remitting course and affects usually young individuals. The prevalence is close to 1% of the population in the Western countries. And it really has devastating effect on their lives. But we still don't know what is causing inflammatory bowel disease. And if you look on the right, these are the factors related to the pathogenesis of inflammatory bowel disease. And inflammatory bowel disease is hypothesized to be a multifactorial disease. Thus, it involves the gut's genetic susceptibility, the environmental triggers, it involves the gut microbiome, altered immune response that altogether contributes to unregulated inflammation and ultimately development of disease. Now, if you look at the recent epidemiology, interestingly, there has been a rapid increase in the incidence of inflammatory bowel disease, especially in the newly industrialized countries such as China and India. And this trend is recapitulating the trend that was seen 100 years ago in the Western world. Now, this suggests that instead of a rapid change in the human genetics, it's likely more the change in the microbiome, in the environment, thus the change in the gut microbiome, dietary changes that may be triggering the development of disease. Now, there's one very important concept when we think about the pathogenesis of IBD. That is, there's a long period of preclinical phase that progresses silently towards a development of symptoms and ultimately a diagnosis of disease. Thus, in genetically susceptible individuals, there's various environmental factors and microbial factors that trigger the inflammation silently, and all this process is proposed to proceed years before diagnosis. But the question is, how do we prove this? And how can we design such a study to understand the triggering factors of Crohn's disease? And one unique way to look at this research question is by prospectively following healthy at-risk individuals until they develop Crohn's disease. And this is the very study design of the one and only Crohn's Colitis Canada Genetic, Environmental, and Microbial Project, namely the GEM project. And this was started in 2008. More than 5,000 healthy first-degree relatives of patients with Crohn's disease were enrolled, and they're followed. They're followed every 6 months by phone until they develop Crohn's disease. And during a median follow-up duration of over 8 years, currently we have around more than 100 that developed Crohn's. And the idea is to compare the baseline biomarkers at recruitment of the healthy first-degree relatives that developed Crohn's disease over time compared to those that remained healthy over time. And this will give us an idea which are the biomarkers that precede the diagnosis and predict the development of Crohn's. Now, we were particularly interested in the impact of the gut microbiome on the pathogenesis of Crohn's disease. And there is currently no cohort that has preclinical samples of the gut microbiome. Thus, leveraging the data from the GEM, we wanted to really look at to see if the baseline gut microbiome composition is associated with future development of Crohn's disease. Now, to assess the gut microbiome composition, we used a culture-independent approach. And this is by sequencing the 16S ribosomal RNA of the gut bacteria. And there's a hypervariable region of the gene that helps differentiate the different bacterial taxonomy. And using the QIIME2 pipeline, we could generate the relative abundance of each taxa at the genus level. And you can see this is a taxa table. It's a high dimensional data set including 165 taxa at the genus level from over 3,483 subjects that had available microbiome data at baseline. And during a median follow-up duration of 5.5 years, 73 subjects, first-degree relatives, developed Crohn's disease over time, while 10 subjects remained healthy. And so with this, we first aimed to see which individual taxa is associated with future development of Crohn's disease. And as you would expect, using a time-to-event Cox proportional hazards regression model for each individual taxa, we looked to see if there's an association with the time-to-event outcome. However, we did not find a significant signal after correcting for multiple testing. And this is likely due to, and we hypothesize that it may be due to the weak individual taxa contribution to onset of disease. And maybe it's the community effect of the microbiome that contributes to development of disease. And so we thought about combining these individual taxa into a risk model. And when we started to think about that concept, we thought that there may be high order interaction between the 165 bacterial taxa, also not only linear effects of the taxa on the outcome, but there may be many non-linear effects of the taxa to the outcome. And in this regard, we decided to use a machine learning approach Here we used the random survival forest model. And this, to a lot of machine learning people, they're probably familiar with the random forest. This is a tree-based model originally developed for classification or regression problems. Recently, this was extended to time to event analyses, and this is the random survival forest. And thus, similar to the random forest models, it has the bootstrapping and aggregating. And so the trees are created, and at each node of the tree, it uses the variables to the variables to split the cases and controls, like in random forest models. But here we include the time component as well, using the log rank test as a criterion. And for each tree, the cumulative hazard function is calculated, which represents the predicted number of events for each tree, and that's averaged. And that's the idea of the random survival forest. Now, what's very important in developing machine learning models is that there needs to be an independent, completely independent validation set to validate the performance of the model from a data set that the model has never seen before. So we split the cohort 1/3, 2/3, 1/3, and developed the random survival forest-based microbiome risk model with the training set, and validated this model in a completely independent testing set. And here is the result from the independent validation set. And this is a survival curve. And the x-axis is the time from enrollment. The y-axis is the disease-free probability. Thus, if you develop cases, if you develop events, it goes down. The group in red are the subjects that are part of the over 50% percentile of the microbiome risk score. So it's a high microbiome risk score group compared to the low microbiome risk score group in blue. And you can see that if you're part of the higher spectrum of the microbiome risk score compared to the rest of the cohort, you're at 2.2-fold higher risk of developing Crohn's disease. And notably, this is the first time that any study showed that the microbiome composition can predict your future risk of developing Crohn's. Now, we were very excited about this result, but we wanted to go a little bit more to understand which variables or which taxa were contributing to this model. And one downside of machine learning models is the interpretability. And what I mean by that is compared to when you develop a conventional regression model, you're able to have the hazard ratio and the confidence intervals of each variable. However, you don't have that kind of interpretability with the variables included in this random forest model. And thus, we need to use other post hoc analyses to try to interpret what was important and what the variables mean. And one way to understand more of the individual variables is to measure the feature importance of the individual variables, individual taxa that contributes the most to enhance the predictive performance. There are several ways to do this. One is a tree-based approach. So the variables that were used that were close, they are closer to the root of the tree are the more important ones. And so based on that position in the tree, we are able to measure a importance and rank order the importance of each of the variables that contributed the most. There's another way in which it's a tree agnostic way. We use the overall concordance index, the C index, and see, and by taking out one variable at a time, we check to see how much of the overall performance decreases, and more you decrease means the individual variable that was taken out was contributing a lot. So there are several ways to understand the feature importance. And for this this is the rank order of the top 20 important taxa that contributed to the model. And some of these taxa were shown in case control studies comparing established Crohn's disease patients and healthy controls, one namely the Fecalibacterium. And this is quite popular in the in the IBD literature. It has an anti-inflammatory effect and is decreased in Crohn's compared to healthy controls. But it's the first time that we see that this taxa is importantly contributing to future disease even before a diagnosis in the pre-disease phase. Now, we can further measure the interaction between any two taxa or any two variables based on the position in the decision tree. The more you're closer to each other in the node means your interaction is higher, and we can calculate that interaction. And this web is based on the top 20 taxa that I just showed you in the previous slide. And you can see that there's a complex web of interaction between the top 20 taxa that's captured in this model. And it probably, and we speculate this is likely that the gut microbiome as a community may be contributing to the future risk of Crohn's disease. Now, lastly, we attempted to go beyond just composition, microbiome composition data, and understand a bit more of the functional aspect of the microbiome. And to do this, we looked at the stool metabolites themselves. We measured the stool metabolites. This was only available in the small subset of the cohort, thus this is more of an exploratory analysis. We used the Metabolon platform, and there's more than 1,000 stool metabolites that were analyzed in this nested case control cohort, a subset of the GEM cohort, where we have 56 cases that developed disease matched to 66 matched controls, healthy controls that remained healthy. And the aim was to see which stool metabolites were associated with onset of Crohn's, which were also associated with the microbiome risk score, and which are associated with both. And so after multiple correction, these metabolites were significantly associated with onset of Crohn's disease. You can see that 5 were associated with increased risk of Crohn's, while 10 were associated with decreased risk of Crohn's disease. And among these 5 metabolites in asterisks were also associated with the microbiome risk score with consistent direction of effect. And we speculate that these 5 metabolites may be mediating the effect of the microbiome composition on future development of Crohn's disease. So in conclusion, what I just showed you is using the prospective pre-disease GEM cohort. I show you for the first time some evidence of pre-disease gut microbiome composition that can predict future risk, individuals' future risk of developing Crohn's disease among healthy first-degree relatives. To do this, I not only used traditional regression models, but also machine learning models, such as the random forest survival model, to integrate the high-dimensional omics microbiome data into a risk stratification model. And I already went over my rationale of why we used machine learning here, accounting for the nonlinear association and the high-grade interaction between these variables. And this helped us to gain insight into the complex relationship and the contribution of the microbiome community on the risk of Crohn's disease. But lastly, I want to mention that association is not necessarily causation, and thus we plan to perform mechanistic studies with germ-free mice models too, and maybe use the findings from our studies and hope that this will guide which microbe or function needs to be tested for causation. So with that, I thank everyone in the GEM project consortium, my supervisor, Ken Kuritoru, who leads this amazing project, And I thank the funders, Crohn's Colitis Canada, Helmsley Charity Trust, and CHR for their support, and Mount Sinai Hospital for supporting my fellowship. And most of all of the participating sites and the subjects that participated in the GEM project. Thank you all for listening to my talk today, and I'm open for any questions.
Thank you, Dr. Lee. That was excellent, very clear, great way to end our trainee round sessions. Can we please open it up at this point for questions, comments from the audience?
I can open while people are thinking. So I wanted to dig into why you had to go with random survival force. So you were saying that basically you expected there may be higher order interactions. Can you maybe speak more to that? What sort of higher order interactions you were expecting could be present that wouldn't be picked up by a linear model like Cox proportional hazards?
Yeah, so I think it has to be more on the 165 taxa that we had was relative abundance. So it means if one goes up, the other may go down. There may be a lot of interaction between these high dimensional data set. Now, I think someone may ask, then why random forest? There may be other models as well. And so we actually looked at other models as well, such as another tree-based model, the gradient boosting survival. There's gradient boosting for survival as well, survival analysis. There's also, we looked at DeepServe, which is an extension of the neural network. I think now all of them had similar performance. The performance was based on the concordance index of 67. Now, the random survival forest model had a slightly higher C index, but similar to the other models that we built. I think one thing that we really like about the random survival forest is there's a lot of ways to prevent or minimize the overfitting. It averages, and even the subsampling and everything. For example, the gradient boosting model, it basically tries to reiterate and increase the performance as much as possible rather than averaging. So we thought that because if you think about it, it's only 2% that develop disease compared to 98% that remained healthy. So there's an imbalance of in the event case and controls in our data set as well. So we wanted to go with a little bit more stable machine learning model. But if you have any other suggestions for other models that can use this survival time to event extension, I'm open to any kind of suggestions.
Honestly, I wouldn't fuss too much about the specific type of model. I meant in principle, a gradient boosting decision tree, if cast correctly, can be a random survival forest. So that's kind of fiddling over the color of the bike shed, but I feel there's something to what higher order information you really want to capture, and the fact that you say it's relative abundance makes me sort of think that you don't really have any. Sorry, I'm just commenting. It's not really a question, but there isn't really a capture of sort of maybe global phenomenon, like do you have a large amount of different bacterial taxa versus, you know what I mean? Like that's not trivially captured in a tree-based model, which I was curious if that could be, not knowing the field, could that be something that's a factor? Just the variety of taxa that you have beyond just you have these specific 2 or 3.
Yeah, I think it's the nature of the microbiome analysis in general. There are some people who are trying to move towards the absolute abundance. However, I think majority of the studies look at the relative abundance. Now there is this type of compositional data. I think it's very hard to, deal with. There are several ways to actually try to transform the data. Let's say, for example, central log transformation in these type of compositional data, and or other ways to try to put this into a Euclidean space. So I think, yeah, I haven't looked at those yet, but yeah, definitely some things to consider in the future.
Yeah, it's not an easy solution. Anyway, thank you.
Thank you so much. There's a couple questions in the chat, and then we'll wrap after that. The first question is from Elliot, and they ask, is there a plan to include other elements you collected in future models? For example, are there any interactions between environmental factors and microbiomes? I think that would be quite interesting.
Yep, that's a very nice question. So this was started about 2 years ago when we had the microbiome data. The next step is, once this is published and set, we are planning to include more data that we have available, such as the inflammatory markers that we have. There's a gut barrier marker that we also have, and some environmental factors as well. We have some diet, and one of the postdoc at our group, actually 2 postdocs, one Mingyu Zhu, who's looking at the environmental data set in our lab, and also Anna Newstater is looking at the diet data, and we're ultimately we would like to combine all of these into a very integrative risk model that can have a better performance than just microbiome risk score.
That sounds really exciting, and it seems like a lot of opportunity there for model complexity to the earlier comment. Okay, last question from Noor. Do you have any plans of determining which communities produce the metabolites that were shown to be associated with CD risk, and how? So yeah, another good question.
Yeah, so I think you mean the function.
Or maybe this is also a bit more about like the population distribution of some of these groups as well.
I don't, can you repeat one more time?
Sure. So do you have plans of determining which communities produce the metabolites that were shown to be associated with risk? Yeah.
Yeah. So so there's several, I think what we want to do, one way to deal with that is looking at the gut microbiome composition that we detected as risk factors for onset of disease, and see if some of those are associated with the stool metabolites. And unfortunately, we don't have stool metabolomic data in every. And I think what I showed you in the previous slide was we had it in 121 subjects rather than 3,400 subjects. So, and this is a pretty expensive data set, especially the metabolomics. We hope to look at this in more detail in the entire, when we have the data in the entire cohort. I think there may be some mechanistic studies to look at this as well, such as you can culture these microbiome and see and apply and do some further in vitro, in vivo tests to see if those microbiome community has a specific function.
That's great. Well, thank you so much. You have a lot of accolades in the chat. Just thanking you for such an interesting talk. I really love this last slide because the collaboration for this type of work is pretty amazing. And I think once you start getting all that data together, there's sort of like an endless list of research that you can do in this area. So thank you very much. Very clear presentation. thanks to everybody for attending. We're shortening a bit today. We typically have 2 talks, and today we just have the one. But I want to close our rounds for 2022. We've had a reminder, we pick the top trainees every year to present and profile at these rounds, and we've heard some amazing talks this year. And we also have the applications for 2023 trainee rounds open. So they close on Friday. If you or someone you know you think would be is working on a really exciting project at the intersection of AI and medicine and would like to be profiled, please encourage them to apply. I'm very happy we're having people join from different areas. Next year, we're actually hoping to see some of our trainees from across the country. So you'll see trainees from different parts of Canada, which will be great. And of course, we award, you're awarded for being selected and then another award for the top trainee of the year. So thank you again for an excellent talk and everyone for joining. Wishing you all a restful holiday and a great start to 2023. Bye, everyone.